Okay, so here is where we left off on Monday. We were talking about transport across the cellular membrane, and we discussed that there were two general categories of transport. There was passive transport, which we see here, and then we also were looking at what we called active transport. Now, the major difference between the two categories is that passive transport, as we said, you will recall, does not require ATP, and I like to use this symbol here. Passive transport does not require ATP, so there's no ATP involved. And then active transport, of course, does require ATP. So here we need ATP to move stuff across the membrane and then get it where it needs to go, and in passive we do not. So then in passive transport, as you will recall, we had two main methods of passive transport. We have diffusion, which of course we also lumped together with, let's zoom in here, I'm going to write it here for you. We lumped it together with osmosis. So diffusion and osmosis were one of our types of passive transport. And then the other type listed down here was facilitated transport. So remember that diffusion and osmosis rely on a concentration gradient. Let's erase this here. You'll see that up top there. Diffusion and osmosis rely on a concentration gradient. Facilitated transport is highly specific, we talked about, and also it's kind of an exchange. So something is happening on the inside. I think let me erase this. I'll stick with the example that we used. Something is happening on the inside of the cell membrane. For instance, you've got potassium going out, which then allows for sodium to go in. So that's an example of facilitated transport, facilitated diffusion. And then, of course, active transport is required in order to move against the concentration gradient. This is helpful for our cells in some circumstances. So remember, in passive transport, we went from an area of high to low. But in active transport, we can move from low to high when we need to do that. And that requires ATP. Sometimes another energy source can be used, but for the purposes of this class, I want you to understand that it's ATP. And then they give an example here of the sodium-potassium pump, which we will explore further. All right, so let's move on into lecture some more. We took a look at these, what we call carrier proteins, which are used in active transport. We also see some protein usage in facilitated transport at times. And we're going to go ahead and skip this animation. Again, these animations are in the PowerPoints that are posted on D2L. So if you want to watch it, you can log in and pull the PowerPoint lecture down, and then those animations should be active for you. All right, so we talked about endocytosis and exocytosis, and that was the idea. If you remember, we go back to that diagram of the organelles within the cell. So we're packaging up stuff in the Golgi apparatus. That's our packaging and shipping center. And we're putting them into vesicles. And remember that those vesicle membranes, the membranes mirror the membrane in composition of the plasma membrane of the cell. So that allows for the vesicle and the cell, like we said earlier, to merge, and then either the contents would come into the cell, which would be called endocytosis, or the contents would go out of the cell, exocytosis. And what this really allows for the cell to do is to bring things in and move things out in bulk. So we're not just moving one of something, we're creating a little vesicle that has, like if you would, a little pocket or a little bubble full of some type of substance, and we're moving it across the membrane through endocytosis and exocytosis. And endocytosis and exocytosis are a form of active transport, usually active transport. And another animation that you guys can watch. Okay, so now let's talk a little bit about actual proteins. We talked about, if you recall, I'm going to switch over to our notepad. This would mirror the whiteboard in our class. So we talked about this idea of the plasma membrane, which has these phospholipids, right? We talked about these phospholipids in class, drew them out on the board. And they form in this manner to make this this phospholipid bilayer membrane. And we also talked about how within this membrane we see like some carrier proteins and such. Some will be embedded in, like in this configuration. Some will actually span the entire width of the membrane, like this. Okay, keep going here. Some may span or not span, but they may have like a glycoprotein or some type of carbohydrate bound to it, and this would act as a receptor. And then our membrane continues, and remember, it's all with that idea that this is a fluid. Remember, we called it a fluid mosaic configuration. So this is flexible, it can move, these can rearrange in different patterns, but they all come together to make the plasma membrane. And then all of these different integrated proteins and etc. have a function in the survival of the cell. So let's go back to our notepad here and continue on. So the ones that span the membrane, which means that they have one side of the protein on the inside of the cell and the other side of the protein are on the other side of the cell. We call these receptor proteins, and they can, they are involved in what we call cell signaling. It's not written on here, but I'm going to put it on here because you'll see and hear a lot about cell signaling if you do any research, or I can write that a little bit neater, or do any reading or Googling onto the subject. You'll want to look at cell signaling. And what these do, like I said, is they kind of act like a scanning barcode. You've got the protein unit out here, and then something comes by and it has some type of a receptor or a unit on it that recognizes the sequence on this, and then they dock, and then this sends information into the cell. Now, the idea here is the idea of a receptor site. We sort of started talking about the fringes of this inside the last lecture in class. Now, here's the idea. Let me flip back over to our jotter. Okay, so if we had this receptor here, there's a binding site, and I'm going to draw it. Let me actually redraw this. If we had this receptor here, there would be a binding site. Let me just do this a little bit neater for you. All right, I'm going to make our protein round for this example, and we're going to have this binding site. It looks like this. So our membrane is maybe across here, and you can see that this protein spans both sides. So this is the extracellular side, and this is the intracellular side. So we have extracellular fluid on this side, and on this side we have cytoplasm in the cell. Okay, and this is our protein. And then I'm going to change the color here. Actually, let me do this in blue. So this blue region here, this we're going to refer to as a binding site. A binding site, okay? And then we'll have some kind of something on the outside here. It could be a ligand or another protein or something of that nature. And it's out here and it's floating along, and whatever its shape, it's going to have this little piece piece here, this little region that is going to basically fit perfectly into this binding site. And this is kind of how they recognize each other. Now, if I have another protein out here, we'll draw it in, let's say, green. If I've got another weird protein out here and its ligand is like shaped like this, you know, it's not going to work in here. It's not going to fit. It's not going to work. It won't react with this transmembrane protein. It won't signal the cell inside to do anything. And it just kind of, that's how we work. So this one here will float around. This one here would float around. There might be hundreds and hundreds of floating around. But this protein on this cell in this receptor is going to be specific for this guy, which we will call a ligand. Okay, something that binds to a binding site we'll call a ligand. So that's kind of the idea of how this works. And so let's switch back to the lecture here. So the receptor sites on receptor proteins, they're saying interact specifically with signal molecules. And then, as we talked about in class where I gave the example of the cord that was knotted up, but then something happened into this protein, and it's still a cord, but now maybe it's shaped differently with this part going inside the cell and this part being outside the cell. And that allows for this protein to either pull things across the cell membrane or send a signal, or maybe this region reacts with something inside the cell that causes something to happen. Just whatever this protein's purpose is. The ligand binds and it changes configuration. And then that triggers something inside of the cell, which is the result of the binding. For instance, let me give you an example. We can talk about glucose and insulin being a really good kind of example for this. And I won't get into the very specifics of this type of protein, but I'll show you. Essentially what happens is you've got this cell and you've got the nucleus here. Now the genetic material, the DNA inside of this DNA, are coding regions that will code to make insulin, okay? So you've got DNA in here which tells this cell how I make insulin. And then up here on the surface of the cell, you've got this protein up here. And then maybe on the inside of the cell, you've got some other little pieces. We'll just call them pieces for now. I'm just going to draw a couple different random things in here that could be attached and working with this protein. And then for argument's sake, let's say down here, you've got this little region here. All right, so this is very simplistic, I guess. I really kind of like made this really simple for you because the details of this are beyond the scope of this class, but I kind of want you to know what's happening here. So you've got this cell, let's call it a pancreatic cell. You eat your meal, okay, and then your blood glucose goes up. Your blood glucose goes up. So now let's say that you've got some elevated glucose molecule out here circulating in the blood. Oh, can't spell right now. Let's try that again. Molecule circulating in the blood. And maybe it looks like this. Okay, it looks like this. Now already we can kind of tell from that configuration and depicting it as a shape as we're doing here that this glucose molecule looks like it's going to interact with this transmembrane protein right here, this signaling protein. And it's going to come and it's going to sit and it's going to dock in here. So now we've got this blood glucose that's triggering this protein here. We'll color that in. And it sets off a reaction. it sends a signal to this, and then this lets go of this. And then this guy comes down here and he instigates some kind of a reaction at the nucleus of the cell, which then triggers the cell to create insulin. Okay? So our glucose goes up, we need insulin, we make the insulin inside of the appropriate cells, pancreatic cells, islet cells, and then we release the insulin in a vesicle out where we need to go. And then that insulin, of course, would go somewhere and talk to some other cell membrane and do what it needs to do, and so on and so forth. So this is kind of how this happens. This is sort of an idea of cell signaling. Now, I mentioned that this is a pancreatic cell. And this is a pancreatic islet cell, and then this is where insulin is produced. But not every cell in our body has the protein to bind and trigger that cell to produce insulin. Otherwise, our muscles would be producing insulin, our brain cells would be producing insulin, our liver would be producing insulin, and that's just not how our body works. So we accomplish this through something called differentiation. And we'll talk more about that when we get into, I think, chapter 4, and how different cells have different functions within the body that they perform. But for this specific lecture, I just want you to know and understand that these receptor sites interact specifically with some sort of signaling molecule, which I called a ligand. And then that ligand triggers a change within the cell, and something happens within that cell. And then here is where the key point that we're trying to get to. Different cells have different receptor proteins depending on the function of that cell, what its job is, what type of cell it is, and so on and so forth. Okay? But this is how we communicate. We not only send molecules across the membrane back and forth, but we also send signals. We also are able to send signals which tell the inside of the cell, the machinery and the organelles that we just finished memorizing, tells the inside of the cell what it needs to do. All right? So let's move down. All right, so this is sort of just another depiction of what it is that I showed you. You'll see here's the receptor site, here's the signaling molecule or the ligand, and it docks here in the extracellular. When it docks, it sends a message, it triggers this protein here to do something on the inside that results in some type of a product or result that we're looking for. And then once we accumulate this product in some kind of a feedback, our body, we'll talk about that later, has mechanisms to know, okay, we've got enough of the product we're seeking. It releases it, then it causes another transformation in this protein, which will release this initial signaling molecule, and then the whole process will turn off. So we'll talk a little bit more about how that start-stop mechanism works, but for your purposes, I just want you to understand that you can have these receptors that don't necessarily bring something into the cell, but they react to some type of outside cell signal to tell it to initiate some kind of action within the cell, okay, within the cell. And so it may be that there are some rudimentary hormones like T3 that are then processed and made into T4, and then we maybe spit that out. Or you may have like some minor puzzle pieces that we can attach things to that become ATP, or we may have some minor players inside of here that we then transform into insulin. At any rate, we're triggering molecules on the inside of the cell to react in a certain way that ultimately results in the production of this product. And that is what the goal is. And at that point, the signaling molecule lets go. So hopefully you understand that. That's really as much as I need you to know about this particular topic of cell signaling, but that is one function of possible proteins. So we're going to talk a little bit, we're going to apply sort of these concepts to one of the main transport molecules, cell signaling kind of mechanisms that we like to study. It's very, very important for human biology, very important for the proper functioning of most of the cells in the human body. And so it's very highly studied, and we know a lot about it, which we don't know a lot about everything, but we do know a lot about this one. So we want to pass it along because it's really just like a reoccurring theme in this class. This is called the sodium-potassium pump. So as we move into this, I want you to take notes and really know how this sodium-potassium pump works, because you will be tested on it. All right, I'm going to pause the video.